Our next speaker is Professor Peter Schmid. He is the lead of this centre, the Centre for Experimental Cancer Medicine, and he's going to chat about new developments in breast cancer. So, over to you.
Hello, everyone. I can't see myself, so I hope you can see me, hear me, and possibly even see my slides. Thank you for coming today. It's really important for us to bring as many people in because all this clinical research wouldn't be anything without us all together working as a team and as a network. Breast cancer, you hear a lot about breast cancer, and you always read there's so much research going into breast cancer. Can we actually still make a difference there? And the simple answer is yes. We have made a dramatic difference in the last few years. And what I'm going to do today is just share with you a little bit where we've come from in breast cancer, and 2 or 3 of the highlights of the last few years, and just 2 or 3 of the themes where we're heading. You will have heard from the others already. There are recurring themes, for example, immune therapy, for example, antibody drug conjugates, and I will pick on that a little bit as well, and show you just some of the developments. Just that we're all on the same sheet. When we talk about breast cancer, it's obviously not just one disease. There's also different stages and different subtypes of breast cancer. The majority of breast cancer have early stage breast cancer, and that's when the cancer is just in the breast and possibly the lymph nodes under the armpit. And in that situation, we have a very, very good chance of curing patients. But we also, in addition to surgery, often give medical therapies, chemotherapy, and other targeted treatments, which we call adjuvant therapy, just to increase the chances for patients to be cured. Unfortunately, in some patients, and it's a small percentage of patients, but in some patients, months or years later, they develop what we call metastatic or secondary or advanced breast cancer. These are different words for the same condition, and it basically means the breast cancer has come back, has spread to other parts of the body, for example, lung, liver, bones, and in that situation, unfortunately, currently we are unable to cure patients, but we have made substantial progress in maintaining the quality of life, but also improving survival, and sometimes substantially. But what we offer to these patients is then serial medical therapies, with each of them with the hope to control the cancer, push the cancer back, maintain or improve the quality of life. So if you look at where we've come from in the last 20 years, is really 20 years ago or 25 years ago, I should say, when I started treating breast cancer patients, we cured about 65% of patients. Now, these days, we are up to about 85% of patients, and we are getting closer to 90%. Again, for patients with incurable disease, with metastatic disease, we have doubled at the same time the survival time, with improving our treatments in a way that we tailor them better, reduce some of the side effects. And so we're not just winning time, but we also, fortunately, are able to improve the quality of life patients have during that time. I've already said at the beginning, not all breast cancers are the same. And if you look at this under the microscope, we have 3 types of breast cancer. We have a large group of breast cancer that has an engine in there that's fueled by estrogen. That's what we call hormone-driven breast cancer. And then we have the other third of breast cancer is independent from hormones. And there's a second big group in yellow here that has a different engine that's driven by HER2. And the remaining group of patients we call triple negative, rightly or wrongly. And that's really relevant because our treatment options for these 3 types of breast cancer are different. For hormone-driven cancer, we try to take the engine away by giving anti-hormone therapy. For HER2-driven cancer, again, we try to take that engine away by giving anti-HER2 therapy. But for triple-negative breast cancer, we struggled for many years to have a targeted therapy. And this is an area where I would say we are particularly good at Barts Hospital. We have probably become the leading research site in Europe for triple-negative breast cancer. And some of the treatments I'm going to mention were developed at Barts and are now changing the way this type of breast cancer is treated around the world. If you just look at breast cancer, how it was treated 10 years ago, and I'm not going to go into the details, just to show you early breast cancer was relatively simple. Patients had an operation, We then assessed whether they were at high or low risk of disease recurrence, and they either received just radiotherapy and hormone therapy, or sometimes chemotherapy and hormone therapy. And those patients with HER2-driven cancers received HER2-targeted treatment. And then about 7-8 years later, 2017, we had already made substantial changes. We are now routinely offering a gene expression test where we do basically fingerprint the cancer to see whether patients are at a higher risk or lower risk of disease recurrence. And that way we can now spare a third of patients from having chemotherapy. And then in terms of patients who are low risk, we now give very different chemotherapy options. Combination chemotherapy, sometimes for 10 years. We're giving drugs that strengthen the bones and also reduce the risk of breast cancer recurrence. And so the treatment options have become much wider. If you now go forward another 5 years, and this is where we are, where we've been in the last year or two, again, the beginning of the pathway is the same, but we're getting additional treatments in, for example, now additional hormone therapies that modify the way we give hormone therapy, and again, cure another third of patients, which we couldn't cure before. Now, one of the big themes, and this is one of the trial areas we have at Barts Hospital, and where we were successful in the past and where we will continue to put a lot of effort in there, is trying to work out, does every patient need surgery first and then followed by chemotherapy, or can we actually do it the other way around? And the last 10-15 years, we've increasingly changed the order. We are now the majority of patients, for example, with triple negative breast cancer, but also with HER2 positive breast cancer, will get chemotherapy, medical treatment prior to surgery, rather than having surgery first and then chemotherapy afterwards. The reason for that is there are two major advantages. The first one is we can make the cancer smaller. If we can make the cancer smaller, we can sort of downstage the tumor and have less invasive surgery. So we have small operation of the breast and small operation of the lymph nodes, which means patients have to suffer less from long-term side effects. The second benefit from giving chemotherapy before surgery is that we can see how well the treatment works. What we're trying to achieve with the chemotherapy is get rid of some of those little cells that may be floating around the body from where the cancer could come back from. But if we take the cancer out, there's no way for us to know and to prove whether the treatment works. Whereas if we give chemotherapy prior to surgery, we can see how the cancer in the breast is gradually getting smaller. And we can divide patients into two groups depending on the response. There's a group of patients where the cancer is completely gone at the time of surgery. We can't find a single cancer cell anymore, even if you take the cancer area out. And you can imagine that group of patients has a fantastic outlook. Whereas there's also a group of patients who still have cancer left in the breast. And if the cancer has survived in the breast, it may have survived elsewhere. And those patients, unfortunately, have a higher risk of disease recurrence. And so what we're using clinically now is we sort of de-escalate our therapies in patients with optimal response, and in patients who still have cancer left, we try to target that residual disease and give a tailored therapy for those patients who need it most. Just to show some of the data we have generated at Barts, this is again in triple negative breast cancer, most aggressive subtype of breast cancer, where you can see here on the right side the percentage of patients where the cancer is completely gone at the time of surgery. And 6-7 years ago, we achieved this in 35-40%. Then we added platinum as a new chemotherapy drug in those patients and pushed that up to about 50%. And a large study we led here from Barts called Keynote 522 then was introduced immune therapy, a treatment you've heard in the talks before that uses the own body's immune defense to tackle cancer. And when we add that, we push the response rate up to 65%. And as you can see, that reduces the risk of recurrence by about 40%. So we cure an extra 40% of patients. But what's really interesting to see is if you look on the right side of the slide, those patients in the two lower curves, these are patients who didn't have full disappearance of the cancer, where you could argue, did the treatment work optimally or not? But actually, if we give immune therapy, even patients who have cancer left have a much better outcome, because we have managed to change the biology and the behavior of that cancer. And that's an area where we will do more research going forward. So where are we heading in early triple negative breast cancer? In the smallest tumors, we are now working actively on on reducing the treatment. So what I described here is de-escalation. We're starting a big study where we give one of those new drugs called an antibody drug conjugate, and tell you a little bit more about that in a second. So chemo-free treatment alone and in combination with immune therapy, and hope that we can spare the majority of patients from having conventional chemotherapy. In the patients with slightly bigger early stage cancers, we have established with our study the new UK, but also the new global standard, and it's now NICE approved, and all patients receive chemotherapy and immune therapy. But what we're looking into, can we optimize this? Can we reduce the amount of chemotherapy some patients need? And with our trials, we are starting for that. We're also starting a really interesting project. Dr. Phillips is going to lead on this, where we look at what we call the microbiome. So the bacteria we all carry in our bowels, because they have been shown to be linked with the benefit of immune therapy. And we want to study this systematically in early triple-negative breast cancer, to see, can we predict who's doing well, and can we predict who are the patients who may need a little bit more. We're also trying to see whether we can shorten the treatment in those patients do really well. But the biggest need is here in this group in red, patients who even after immune therapy still have some cancer left. And this is where we started a number of new studies. One, for example, Tropium Breast 03, really exciting trial where we bring a new antibody drug conjugate, a very targeted therapy in there, in an attempt to improve the outcome of patients in this situation. Now, when we look at metastatic cancer, so secondary advanced breast cancer, as I mentioned before, we try to prolong survival and improve the quality of life. And you're aware of the three main subgroups of breast cancer we have. Our research at the moment focuses in patients who have hormone-driven cancers on understanding hormone resistance, because over time, every cancer will develop resistance to the therapy. And we are developing new drugs that go around this and improve the effect of hormone therapy. And in HER2, again, we're bringing new drugs in, and there's a big development, and it's a recurring theme on antibody drug conjugates. And in triple-negative breast cancer, well, I'll talk a little bit more about, we have developed a number of new targeted treatments we have already established and we will establish. Just to show you how complex things are, I already mentioned triple-negative breast cancer is a subtype of breast cancer. Now, what you see in front of you are the subtypes of triple-negative breast cancer. So triple-negative breast cancer isn't one group. There are 4, possibly 6 different groups of triple-negative breast cancer, and we are starting to understand them differently and starting to treat them differently. On the right side, you see the novel treatment strategies we are applying, especially immune therapy and especially antibody drug conjugates. And immune therapy is again something for advanced cancer that was largely developed at Barts Hospital. There are 2 large studies for patients who have metastatic incurable triple-negative breast cancer who received chemotherapy with and without immune therapy. And as you can see from those curves, is patients who receive the combination of immune therapy have a substantially better outlook, live substantially longer than patients who don't have immune therapy. And that has become the global standard. But the next, I would say, 3 to 5 years are probably focusing on a slightly different group of drugs. And these are antibody drug conjugates, and you've heard about this before. And basically, if you look at the antibody as a part of the immune system, that's what looks like this big Y. It targets surface structures on cells. And we found antibodies that find cancer cells, triple-negative breast cancer cells or hormone-driven breast cancer cells. But these it's a bit like a key that goes to a cancer cell, but rather than just binding to it, it has chemotherapy glued to the end of it and brings it straight into the cancer. And this is how the whole thing works. The drug, or that's how we thought it worked in the past, the drug binds to the cancer cell, gets taken up in the inside, the antibody gets broken down, and this chemo, this Death Star in red, gets released and can destroy the cancer cell from the inside. But we have improved the way these drugs worked over the last 3 or 4 years. And now we've learned that if you use a specific type of drug, the chemotherapy drug, it doesn't just stay in the cancer cell, but can also be released in the other to the other cells that just adjacent to the cancer cells. And that's what we call a bystander effect. And that's really important because many cancers, not every cancer cell looks the same. So if you look here, you have some cancer cells in pink and some cancer cells in blue. And let's say the pink cancer cells are the cells where the chemotherapy drug, this ADC, antibody drug conjugate, can bind to. In the past, it would have only destroyed the pink cells. But now that this drug can also destroy the other cells around it, it will destroy the pink and the blue cells, and therefore has a substantially higher activity, as we see in many examples. And for example, in triple negative breast cancer, but similarly in other types of breast cancer, there's a whole range of these drugs coming through with really encouraging, really promising early data. And some of them have already become standard of care. We've also learned that with this novel technology, we can target different group of patients and tumors where we previously didn't have benefit from such drug. I mentioned HER2 positive breast cancer. And in the past, we only called HER2 positive if this HER2 engine is really highly expressed on those cancer cells. That's what we call 3+. And that was based on the old drugs we had. They only really showed a benefit in patients who had this very high expression of HER2. Now, with this new generation of drugs we have, with these new antibody drug conjugates, we can, even in patients who have a lower degree of HER2 expression, see substantial benefits, substantial improvement in outcome. And that's 2/3 of patients with hormone-driven cancer. That's 1/3 of patients with triple negative breast cancer. And so we're defining a new subtype of breast cancer called HER2 low, where the treatment options are being improved very quickly. And just to give you one example for triple negative breast cancer, how we're changing the treatment, you see on the left side, the treatment for early breast cancer, where we have the new standard of chemotherapy and immune therapy. But in a metastatic disease, we now have a number of different options. You see in pink, we've introduced chemotherapy and immune therapy, and we have this first antibody drug conjugates entering in there. But what we're achieving now is bringing new combinations in. One of the areas we're researching a lot at Barts is combining these new antibody drug conjugates with immune therapy. And we see data from 2 of the studies we ran at Barts Hospital, where we could actually show that if we had immune therapy to antibody drug conjugates, we have an activity we have never seen before. So to put this into context, you see on the left side, chemo-free. It's an antibody drug conjugate called Dato-DXd, and durvalumab is a form of immune therapy in metastatic triple-negative breast cancer. Now, the chemotherapy would work in about 40% of these patients. What we saw here, that it worked in this small study of patients in nearly 80% of patients. Similar on the right side, again, chemotherapy would have worked about 40-45% of patients, and we see a response at 60-65%. So we are not just making the treatment more targeted and therefore hopefully better tolerated, but also we're making the treatment substantially more effective, and we're moving these drugs now forward. So one of the areas where we are looking into is how we can combine those different antibody drug conjugates, for example, with immune therapy, but also how we sequence them in triple negative breast cancer. And there's a number of studies we have ongoing at Barts, and some of them we lead globally, which I'm convinced will change the way we're treating breast cancer. But we're also moving these antibody drug conjugates not just into triple negative breast cancer, we've also moved it into the much bigger group of patients who have hormone receptor positive breast cancer. And there, I'm not going to talk much about hormone therapy and hormone resistance, although I could talk about this for 2 hours, but I'm worried you will fall off the chairs. But where a huge need is in those patients where hormone therapy does no longer work, where in the past we only had chemotherapy. These are a group of patients we call hormone refractory. And we would give one chemotherapy after another for as long as these treatments work. Now, we've learned that we can now give antibody drug conjugates there as well as a targeted therapy. And with these treatments, we substantially improve outcome. We have already demonstrated that we have 2 new drugs that improve survival of patients in this situation, and therefore completely changing the way these patients are treated. But this is not where we're going to stop. Now that we've established that these treatments work in advanced breast cancer, our next step is to bring those treatments forward, and we try to incorporate them into early breast cancer. Now, a very complex slide. That's a trial concept we are trying to get off the ground at the moment, where we look at patients who have low to intermediate risk or intermediate to high risk early hormone-driven breast cancer. And then we do one of those fingerprint assays I mentioned to you before. And if that assay gives us an intermediate result and patients don't respond well to a short trial of hormone therapy, we want to give them, rather than chemotherapy, one of those antibody drug conjugates, which in the hope that although these patients need more treatment than endocrine therapy, that we can hopefully spare them from chemotherapy. So you can see the development is becoming increasingly complex. Now, if you go back to our starting point, the 65 to 85% cure rate, the doubling of the median survival time, was achieved without all these latest advances I just spoke about. You can imagine with these highly effective, highly targeted drugs coming through, that we are going to continue to improve outcomes for both patients with early breast cancer, but also for patients with advanced breast cancer. At the same time, we try to reduce the side effects, we tailor the treatment better, and therefore improve or maintain quality of life. Now, I often get asked, what's the next big thing? What's the major leap we are going to see? And I don't know the answer to that. But what I would say is the biggest achievements we achieve is not just by making one big leap, but by incorporating little steps, incremental steps that, if we take them together, have already led to a significantly better outcome in early and advanced breast cancer. The downside of that is the treatment options are getting increasingly complex, and that obviously gives us as choices and challenges for us. So there are increasing demands to the research team, but also to the clinical teams, to explain this better, to communicate these different treatment options better. And we really need an optimized team to help patients to make the right choices in this increasingly more complex landscape. Thank you very much. That's what I wanted to share with you on breast cancer. I'm very happy to obviously answer questions.
Second, we're muted here.
Peter, can you hear us?
I can hear you. I can even see you.
Good.
Perfect.
Amazing.
Yeah.
I have a general question. For a lady who has had breast cancer at her early 30s and never had any children, is there a hope for her to have a baby post-cancer?
It's a really important question. So we just actually, there was a fantastic groundbreaking study that came out a few months ago called the Positive Trial. And this was exactly in that group of women you described, young women, premenopause, who would like to have more children. And these are all these women had hormone-driven cancers, where until now we would normally give hormone therapy for at least 5 years. But if you're 37, you may not have 5 years if you want to pursue your family. And so what we did in this trial is, after 2 to 3 years of hormone therapy, if patients were cancer-free, then patients had a treatment break and tried to obviously pursue their family plans. And fantastic results in that study was led by Anne Partridge from Boston. Out of 500 patients, 74% of patients managed to fulfill their dream of having children. So 74% of women had children. The children were healthy, so there was no difference in the health of children. And most importantly, well, not sure about it, it's most importantly, but equally importantly, the risk of recurrence in those women was not substantially increased. When we compared it to other studies, what we call historic controls, actually it seemed to be as least as good, possibly a tiny bit better. So we have now established a new standard with this research study, which was published in the New England Journal of Medicine a few weeks ago, if you want to read it up, and can now give patients just much more reassurance that if they want to expand their family, it seems to be relatively safe to do, which I think is critically important.
Thank you, sir.
Thank you, Peter. Thank you very much for your presentation. It's been lovely. We'll see you. Have a lovely day.
Thank you, everyone. Bye-bye.